All right. Thank you. Yeah, we'll go. Hello, folks. Oh. Hello, everyone, and welcome to another one of our TESS seminars. And you already know what that stands for because I say it every single week, so I won't say it again. But TESS, of course, is one of our main forums for terrestrial research here at JCU and aquatic research, not marine research so much. And we're really pleased today to have Peter Banks from the University of Sydney here with us. Peter works in animal behavior, in feral animal control, and a number of other sort of synthetic things, cross-cutting things. Peter was trained at the University of Sydney, both undergraduate and PhD, and also with CSIRO. And you probably know University of Sydney is possibly Australia's best university. If it's not, it's in the top 3 for sure, but it's an absolutely outstanding institution. And Peter then went on, he spent, I think, about a decade on the faculty at the University of New South Wales, and now he's a professor at Sydney. So we're looking very much forward to having Peter telling us about messing with their minds, urging sensory tactics to manage pest impacts. And I'll just mention, Peter, you're going to have quite a few people following online as well. It's not just this group here. So thank you very much, Peter.
Yeah, thanks so much. Thanks so much, Bill. Thanks for the invitation. I'm absolutely delighted to be here. I admire the work that's been done at JCU. I was only talking about your research in lectures a couple of weeks ago, actually. So it's an absolute pleasure to be here and come up for the overnight to talk about some of the work that we've been doing. Annabelle's invitation was actually, I'll just check that it's just down, is it? Across, down, click on it. Down, up, space. Arrow, up, down. Hold on. Try again. Here we go.
Yeah, just show it.
Good to go. Yeah, so Annabelle emailed me and said, would I come and talk about a paper that we had just quite recently, which was actually about interactions between a couple of different rodents. And this is the video that was the basis of that paper. And I think it actually captures what I want to talk about today, which is the significance and the importance of behaviour when it comes to wildlife management. So in the foreground here, we have an introduced black rat. It showed up at a spot. It's visiting one of our devices. It's showing that it's there. That's how we monitor whether these things are in the ecosystem. You might spot, though, in the top right corner, a couple of little glowing things going there in the background. And 3, 2, 1, that was a water rat. Launching an attack. So the water rat is a native species. It's our largest rodent. And we wrote this very short paper about that particular observation. But unfortunately, that's all that paper was that observation, because it was relatively unique. But it does capture what I want to talk about, which is the importance of behavioural interactions. The rat showing up at the device was all about making a decision to be there and respond to the information we put out. The behaviour was all about how the two species interacted with one another. And so a lot of my work, as Bill said, is all about trying to manage wildlife and do wildlife management. And I think that it's all about managing their decisions. We rely on them to make decisions to do what we want them to. So if we want to detect them, if we want them to eat baits to control them, if we want to trap them, it relies entirely on them deciding to do it. Entirely, it's their decision. We hope that they will, but it's their decision to do it. And similarly, when they eat grains, when they prey upon birds' nests, when they eat plants that we don't want them to, again, it's their decision to do it. And so managing wildlife, it's traditionally been about just killing them, but even trying to kill them involves the decision that they've got to make. So it's all about making decisions. And so we've been trying to work out, well, what drives an animal to make different sorts of decisions? And we think it's broken up into a number of different components. So the probability of interacting with some sort of device is related to the value of the stimulus, like what are we offering them to be there? It's also related to the perceived danger that they might have of interacting with the device or showing up at that spot. But there's an extra one, and that's the missed opportunity cost. That is, what else could they be doing with their time, or what else could they get without having to take the risks? Or maybe they'll be able to get something better. And so it's these combinations of things which really drive the decision. And today I'm focusing on this last bit, because for years people have been trying to work out some sort of super attractant that's going to draw animals in, or some sort of device that'll be the ultimate killing thing. They haven't really focused so much on the last part, which is the missed opportunity costs. You made a decision today to come and hear me and factored in your missed opportunity costs. You decided to come here and spend your time in an hour with me. You could have been working on emails. You could have been checking social media. You could have been having an early dinner. You could be out exercising. So you're experiencing potentially missed opportunity costs by being here, and I hope that they won't be too high from my talk. Animals are making the same sort of choices. So this is just like a schematic of a rat making a decision about entering a device. It might be attracted from some distance away in T1. At T2, it's decided to perhaps approach the trap. In T3, they really may not go into the trap at all. We want T4. We want the sort of close interaction to take place. But what we've been doing is actually working with this missed opportunity cost idea and putting it into a Bayesian updating framework, because of course animals are constantly making decisions based on their prior experience. So what they've experienced before influences what they'll do next. So in a classic Bayesian updating framework, an animal comes to a situation with some prior knowledge of what it might get or what the value might be. It has a new experience with something, and then what that experience is goes into influencing their prior state next time, but they'll make a decision based on what their previous experience was. What we've been doing is playing around with misinformation, trying to manipulate their perceptions of missed opportunity costs to change their behaviour so that they update their prior with experiences that we give them, so that the next time they come to experience that information, they think that the missed opportunity costs are really high, and so they either won't do it or they might do it. And there's lots of fodder here. So in this talk, I'm going to go through three examples of how we've been using misinformation to manipulate Bayesian updating strategies to address animal decision making for good purposes. The figure there is trying to capture the olfactory world, because that's the world that we work in. Our research is all about the odours that are in the environment that manipulate and change and motivate animals to forage. The three case studies are trying to stop mice from finding wheat grains. The second one is to try and stop wallabies, a native species, from getting seedlings, which is really important in regeneration. And then the final one is about trying to actually catch animals that are really hard to catch because they don't want to get caught. And this is all work of students. I'm just in the background. No, I participated in many of these things, but this is all the work of my research students. And there's Finn Parker, who just defended his thesis earlier this year. And here we're trying to stop mice from causing this problem. It's not super clear in this picture here, but you'll see it perhaps in the next video. Yeah. So wheat is Australia's most valuable crop. It's worth $12.5 billion. The seeds are sown just below the surface, 2 to 3 centimetres, but you can kind of see in this footage here, those holes that are there is the mice just going along and picking the seeds out. They know where they are. They know exactly where they are. They forage with ease. That other figure there you can kind of see. They're not foraging through the soil. They're just going along and picking them out. It's only a 1 to 2 week period when this happens. And the idea here is, can we use a form of olfactory misinformation to manage that? And I'll come back to why we want to do that. And that's because the approach has been just poison. Let's just kill the mice. Put out poisons to try and reduce numbers in the hope that'll reduce damage. The problem is that the poison we use is pretty much banned, which is zinc phosphide, banned in most places in the world. It doesn't really work because it's not strong enough, and then the Poisons Authority won't allow a much stronger poison to be registered yet. The poison is incredibly toxic to birds, and there's growing concern globally about the increasing residues, etc., from secondary poisoning. And yet mice plagues cause, the plague we had in 2021 caused $1 billion in damage. So we need alternatives, and we need things that potentially work. And this is what ecologically based rodent management is all about. And we work with CSIRO on this. We want to not use as many chemical compounds to kill things. We want to manage the impacts, not the species. And really, we want to manipulate behavior. And so this kind of idea really fits in with that quite well. So rodents are olfactory foragers. Ratatouille is my favorite movie ever. So we started off this work trying to understand how they navigate their environment. And we've done experiments putting out odors. This is where we're seeing are odors of birds' nests what caused them to go and visit them. Only a tiny amount of odor put out of the nest, and the rats are there within a day or two. This is putting out quail odor. The rats are visiting them within a day because they're navigating the world with smell. And really, we wanted to stop this. This is actually where the work started. We wanted to stop this problem because we put out odors of all sorts of species, and every time the rats would visit there, just putting out the amount of bat poo produced by one bat on one night was enough to increase visitations by rats. That's how sensitive their noses are. And there was a paper recently about rats attacking microbats, which is really cool. So how has nature solved this problem of being conspicuous to your enemies? Well, through camouflage. And these birds here are a great example. I think you've got the bushstone curlews around here, don't you? Yeah. You don't have kakapos though. Well, these are both beautiful examples of visual camouflage. You know, animals blending into their background through selection so that they're not as visually conspicuous. It is much trickier with odor, way trickier. So visual camouflage, evolution is selected to match the background. You can't really, well, you can, but we won't go there. It's very tricky to not smell. And in a management sense, though, what we thought we'd do is we just flip it around. Instead of trying to make the organism not smell, we would make the background smell exactly the same as the organism and create a kind of olfactory camouflage. So this is because, you know, when there's a nice tight association between the cue and the reward, and it's really distinct from the background, you get these receiver operator curves, which is kind of like the way to show how precise can an animal forage. This is when an animal's foraging really precise. You know, the chances of getting it right, the chances of getting it wrong are really distinct. What we're trying to do, though, is make it way more like random so that foraging around the cues that just aren't useful for them, then their missed opportunity costs go up and it becomes not worth their while. Even though they might solve the puzzle because they've got incredible noses, they won't put the effort in because they've got to do other things. And that's really what the idea is like. We're trying to make the targets unprofitable for them so that they will just forage for something else. And they should rapidly learn this. So I am getting to wheat, but we actually started this work many years ago, almost 20 years ago, playing around with mice in enclosures where wheat was growing all around us, but we didn't actually work out what to do, but I'll come back to that. We were actually thinking about, can we stop rodents from finding, you know, prey? And so we did these trial experiments, and it's a good way to sort of show the concepts behind it. In the patchy scenario up the top there, that's what it's like when a prey stands out in its background. We put peanuts in a little petri dish with sand in there, and we put some peanut oil, trying to simulate the cues that are being put out by prey. Really conspicuous. That's the patchy scenario. The camouflage scenario down the bottom of this, we just put that peanut oil everywhere so that all the patches, all 100, not just the 15 that had prey in them, smelt like the cue. And so really we took away the rodent's ability to find where things were. We did it in a patchy scenario and random, and this kind of played around with the strength of the signal to background ratio, where we had really weak in the camouflage up to the strongest scenario. And what this showed us was that whenever you make it hard for them, even when a random or really even diluted, mice just give up. They go and forage for something else, what they were always foraging for, because it's just not worth their while. And so you can see the dotted line there is foraging at random. When it's patchy, they can forage with great precision and very effectively. When you make it a little bit hard for them, it just goes essentially to random. So then we took this into the field. Actually, the first time we took this into the field to try and protect birds' nests from rats, it completely failed. We just attracted rats to the site and they ate all the eggs. Artificial nests, we didn't kill any birds. But then we did it on a bigger scale, and then we used it with, we used the camouflage treatment, but we actually added an extra one, and that was tapping into the learning concept. We put the odors out everywhere in the environment before we put the artificial nests to try and teach the rats that if you pursue this smell, it's not going to be rewarding. And so we were trying to really tap into that Bayesian updating idea here. But we also had the camouflage treatment as well. But it turned out that the actual training was the thing that really worked in this scenario. And in fact, it increased the survival of the nest by 62%. And it really did reduce the amount of visitation the rats were doing when they'd been pre-exposed to an unrewarding cue. A chance encounter with a colleague in New Zealand at a conference. I talked about this and he got super keen because there they have problems with stoats and cats and ferrets and rats raiding the birds that breed on the braided rivers in New Zealand. They build very camouflaged nests, camouflaged against their native avian predators. The eggs, you can hardly see them. However, they've got a whole bunch of mammals that hunt differently. They hunt by smell and they can't control them. They can't kill enough of these predators. And the bird species like dotterels were all in dramatic decline and they couldn't control the predators. So they were willing to trial something else. And so what we did was we scaled this up and we created artificial odors and put them out in really big sites, several hundred hectares, put out hundreds of odor points in 2 treatment and 2 control sites. We put cameras out and monitored what was showing up, etc. And then flipped the treatments in the 2nd year because our colleagues there knew that no one would be convinced by an N=2 experiment. So flip the treatments and to see what would happen. When we put out the odors, the predators did indeed show up first, but then they lost interest. All 3 lost interest. The hedgehogs were a bit late because they emerged from hibernation a bit late, but you can kind of see a rise and then a fall in all of them. They're showing up because they're interested, but then they stop showing up because they lose interest, which is what we predicted would happen. And that led to way better survival of breeding birds. In fact, 70% increase in hatching success and fledgling success that they modelled. And I would suggest that if we kept it up, we would turn declines into increases. And that was without killing anything. Okay, back to mice. So we knew that it would work there. We hadn't actually thought at all about the idea of actually going outside of the enclosures where we'd started all these experiments to go to the paddocks where this sort of damage was being caused. We didn't even realise it until I saw a picture like this where I saw them pulling the seeds out. So we just thought, can we use this same idea to stop them from running along and picking the seeds out? We know that the mice were attracted to the smell of the wheat from the wheat's germ. It's full of wheat germ oil, which is in lots of cosmetics, etc. And the mice are super attracted to that. Here's a little video of a patch of wheat germ oil in the dark there, and the mice running over and going, that's where there's got to be wheat buried, because it's the cues that they use. So the idea was, could we put that smell everywhere, stop the mice from finding the seeds? And so just a schematic here of how it's working. Seeds in the ground, emitting odours. Mice can find them super easily. The solution, put that same odour all over the surface. Mice shouldn't be able to work out where the seeds are. So we did this. We wanted to do it for a number of years, but it was really challenging because there were no mice anywhere. But then in 2021, there was a mouse plague, which then we had too many mice, but that was better than nothing. So we did this on a paddock where a farmer was happy with us. He was happy to not poison the area, which is really good. We had small plots, which were 10 by 10 meters, but that's about how much a mouse moves during plague conditions. There were very many mice, and it was just, the paddock was just sown, the farmer just did their thing, and then we did our thing. And this is just an aerial view of the sowing lines. So we had 4 treatments. We pre-exposed the mice to this wheat germ oil so that they got used to it and realized that it wasn't worth their while. We had our camouflage treatment where we put it everywhere. We used canola oil as a oil control. We had a trampling control where we just walked over the plots as usual in case we affected the seeds themselves. And then we had our untreated control. And I was the photographer here because I was out there counting seeds and etc. with them. Doing it a stupid way, we realized afterwards, we're not farmers, we realized that there is much better ways to spray things than using hand pumps that you buy from Bunnings. But anyway, yeah, so we counted how many seeds were left after a week, how many seeds were taken after a week, how many seeds were taken after 2 weeks, and this was right up to the point where they started to emerge. Might just skip through that, save time. Did it work? Indeed, it did seem to work. You can see from this figure that where we applied our priming treatment and our camouflage treatment, they were pretty much the same, but they were significantly lower than the control and all the controls. And in fact, after 2 weeks, after 14 days, there was a 73% reduction in the damage that mice were causing compared to when we used our control plots. And then this led to more seedlings emerging. The mice did seem to learn there was more damage in week 2 because they're not normally digging in the ground for seeds. They're normally just eating stuff on the surface, and they learned that there's stuff in the ground, and that's why the damage was bigger in the 2nd week than the 1st, but our treatments seemed to stop that. And so, yes, there ended up being more seedlings on the plots, I think about 80% or more. So yeah, it's really exciting when you can confuse a mouse because they're very smart animals, and we managed to confuse them after 20 years of trying to do it. We ended up with way less damage. So the damage was about 11% of the crop, and we reduced that down to less than 3% based on the sowing rate was there. How does that compare to baiting? We don't know. It's actually not ever been measured about how much reduction in damage you get from putting out poison, because it's quite hard to work out. I'm not throwing shade on people who try and do this. It's just really hard to get. But at least we know that ours works. We think it works because it works with the animal's motivation. They need to forage to find food. They can't spend the time working out these puzzles. They'll just eat what's the easiest option for them. It's using the information that they use to forage. So we don't think that they can really easily learn it. They could smell through it, and clearly some did, but we just think it's not worth their while to do it. And it's using a relatively cheap byproduct of wheat. So now we're working with the CSIRO to try and scale this up. They work on the mouse plagues. We want to work out when do farmers need to use it, how can they use it, etc. And then can we protect other crops with it? So I had an honours student this year working on canola. And obviously farmers want to know what's the minimum amount they can use because they don't really want to use poisons, but they want to know what's the minimum amount they can use to get an effective result. Okay, the second example, another PhD student, this is Pat Finnerty, who is a postdoc at Sydney at the moment. His PhD was all about trying to use these same broad concepts, but now to stop a native problem animal from causing problems to native vegetation using olfactory misinformation. I think probably many people know that herbivores can change ecosystem dynamics quite a lot, particularly by munching on seedlings. This is work done with Claire MacArthur, who's also at Sydney Uni. Her career has been a lot about trying to understand what motivates herbivores to make the decisions they do. And she's been working in Tasmania where they use lethal control and they shoot possums and so forth to stop them from, and paddy melons, to stop them grazing on seedlings in areas after forestry. So is this olfactory misinformation a better option than traditional methods? So this work was done north of Sydney, looking at the swamp wallaby, who's our target animal, and Claire's worked on that for many years, foraging on a eucalypt species, which is highly palatable to the swamp wallabies. So the first was all about trying to tap into this idea of associational refuges. And I think probably, that'll come back to that. First thing we did was, we did the same sort of experiment. We put out odours of eucalypts around a eucalypt in a relatively big area to see whether or not the concept of camouflage would even work at this kind of scale. So we buried vials with eucalyptus leaves so that it was unrewarding. The animals couldn't get access to them. And so they would visit many of these patches and find out that the cues like that weren't useful. And we put them all around a real seedling. This was done by Laura, who did it for an honours project. They certainly visited the unrewarding odours. They did definitely visit them and investigate them. That's what we expect them to do. It's a cue for them to forage, so they should go there and visit them. And they did certainly show up. And there was not really any difference in the visitation or the investigation. But for some reason, and we're not 100% sure, and the p-value is not huge, they didn't browse for seedlings when they were there. So they showed up about the same to individual plants, but they just didn't eat them when they were there, which is quite puzzling, and I don't really know why, because they shouldn't have shown up, but they did show up. It's just that they must have made some association that the cues that were coming out of the plant with what they'd experienced in the ground and confused them in some way. But it did seem to work. So this was, and we're trying to repeat this now to try and understand what was going on, because the camouflage idea didn't lead to a reduction in visitation, but it did lead to a reduction in damage. And so we're going to repeat the experiment to see if we get the same results. And we certainly want to know whether or not it's going to last in the long term. The second part that I was going to talk about that I introduced this before was, can we use misinformation in a way to change how animals forage on that bigger scale? So can we actually put into their heads that a patch is not quite right for them? And this is where the idea of associational refuges comes in. So associational refuges is where plants that grow together. If you grow with an unpalatable plant, potentially you'll have lower rates of browsing because animals will perceive the smell of the unpalatable plants on the wind and not go to those patches because it'll be full of things that they don't eat. That's kind of how associational refuges work. And so there's lots and lots of examples everywhere of where plants growing with an unpalatable plant can have better survival. If we want to use that approach, though, in a management sense, is problematic because, of course, the unpalatable plants compete for sunlight and compete for water. So it's tricky to do that. So the idea that we had here was, can we kind of replicate this associational refuge, but through odours? Can we use the odours to change the way that the animals perceive things? So we know that physical refuges potentially work. Can we create virtual refuges just by using the odours? That'll change the animal's perception of the value of patches, again, change their perception of their missed opportunity costs so they don't go to the patches that smell unrewarding and they go somewhere else. So same foundational species, same problem, etc. Wallabies love that species. And it's usually very hard to stop a motivated animal from foraging on something. You can put a fence around it, they'll destroy the fence. So this is a kind of sneakier tactic. The tactic that we're using is the smell of this plant, Boronia pinnata, which is a really smelly plant, for us at least. It grows in spots where it doesn't get anywhere near as much wallaby activity as places without it. It seemed to offer the option for a good associational refuge. It smells bad. That's probably the cue that the wallabies are using in their foraging decisions. So first of all, Pat did an experiment where he put out and tested the associational refuge concept with real plants. 5 plants around one individual seedling, 1 plant around an individual seedling, 1 plant by itself with no refuge there at all, or procedural control with just the pots. And unsurprisingly, if you're unprotected seedling that he put in the ground that smells great and exactly what you want to eat, they just last barely 2 weeks out there in this environment. But when it's surrounded by plants that smell like spots that wallabies don't normally forage in, the associational refuge idea, you get way better survival. That's with real plants. So then Pat's idea was, can we then recreate those odours that are in these real plants? So is it going to work? So what we did, and this has been a bunch of work that we've done for a while, we're trying to work out what are the odours that are involved here? What are the odour signals? We're not chemical ecologists either. We're not farmers, we're not chemical ecologists. But we do work with a chemical ecologist who knows the chemistry really quite well. And so we've been capturing the odours of things around plants in this case, putting it through a gas chromatography, mass spectrometry, working out what the volatile odours are coming off. And Gabrielle Orlando came up with a new way of trying to work out what's the information that's coded in all of these compounds. Because you could have like hundreds and hundreds of compounds that are there, but for it to be useful to a wallaby, they've got to be relatively consistent. That is, all the plants have got to have these compounds in them. So we came up with a way of actually working out what that is, which is quite useful. In this case, it was just 7 volatile organic compounds. And essentially, that's what's consistent amongst all the individual plants of that particular species. That's likely to be the signal that we can then try and recreate. And that's what Pat did. So then he did an experiment where he put out a seedling by itself, he put out the procedural control, he put out real neighbours, he put out just the odors using this recipe that we'd worked out based on the volatile compounds in the right sort of combinations. We put out another treatment where the combinations were mixed up so that they weren't in the right ratios together. And then we had a whole mix of grab bag of other compounds that were there as another kind of procedural control. And super excitingly, it did seem to work again. We got really poor survival of those unprotected seedlings. The wallabies just went in there and smashed them. But when the odors were out there in the environment, whether they came from real plants or our virtual plants, the survival was still low. Like, it was still low. They were still stumbling upon them and eating them, but it was way, way, way better than the survival by itself. In fact, it was 20 times better. So the actual refuges definitely work, and they seem to be as good as these virtual refuges when we can get the compounds right. So, you know, again, it's another example of animals navigating their world through olfaction, but then us being able to manipulate that information, that olfactory information, to just change what they think about the world and just nudge them towards doing something else, nudge them in another direction, nudge them towards something that we want them to do or don't want them to do. And so now we're working with people in Tasmania to try and address that problem of wallabies eating seedlings down there. Like I said, the Tasmanians, they shoot wallabies, they shoot paddy melons, they've got a license to do it because it's such a problem for them. So we're trying to work out whether or not we can use these odors, different odors down there, because the wallabies forage on different things and they navigate the world with different odors. And we're also using it to try and stop wallabies from getting threatened plants, where the plants are out there by themselves, they're unprotected. Can we use this virtual refuge idea to try and protect endangered plants? And I've yet to have anyone interested in trying to use it in gardens. I've been speeding through, I realise. Sorry, I'll slow down. The last example is another example of olfactory misinformation. And this is where we're trying to trap animals better, basically. It wasn't until we actually had wildlife cameras that we knew what went on at traps. We just put traps out and you wouldn't catch anything. You'd think, oh, there's nothing there. Damn. But then with wildlife cameras, all of a sudden we started to see this, where an animal would show up and just wouldn't go in. And if we put out, in New Zealand, they use the devices on the right there. They're called tracking tunnels. They just measure an animal going in and leaving footprints behind. Again, it's the same thing. The animals show up, but they don't go in. They're suspicious of the devices. This is the PD in that equation. They don't go into our devices because they perceive them as too dangerous. No matter how good the attractant we put out, they really don't like going in. So again, can we play around with another lever in this equation, which is the missed opportunity cost, to try and nudge them to get them to go in? And this is sort of going back to the idea that all the reward, whatever we create, it's all relative. It's the decision, any reward that we might come across, it's all relative to other opportunities. This was part of Finn's PhD again, and I think he did his whole PhD on the concept of missed opportunity costs and how decisions are all relative. And I think it might have changed his mindset quite dramatically, in that now he views everything as just relative to what else he could be doing in the world. But anyway, he's surviving okay. Like, people who try to trap animals already know this. If you try and, if you've got animals in your backyard that you're trying to catch or get rid of, and there's enormous amounts of food everywhere, you'll never get them to go in. And that's because they're missed opportunity costs. Their perceived of interacting with your device are just too high. They don't have to. There's food everywhere. So they're really hard to trap. Whereas if you're doing rodent control, what people will do is usually reduce any other available food that's around. That way you're much more likely to get some sort of interaction. That's because the missed opportunity costs of interacting are much lower. Pretty simple. We know that animals won't interact with our devices. This is some work that we did where we caught animals in one device. We mark them And then we looked at how they responded to other devices, different types when we put them out. And we had cameras on all of them. And we left them out for 10 nights. And we only barely got 50% of them actually going into our device. They just wouldn't go in. It's a persistent problem. It's not a trivial one though, because it affects eradications around the globe. People are trying to eradicate rodents from islands. It's a major initiative everywhere. It's working a lot, but when it doesn't work, it costs an awful lot. So Lord Howe Island, they recently did their rodent eradication. They spent $17 million to get rid of 200,000 rats, about $85 a rat. Unfortunately, 2 rats were detected 2 years later that weren't reinvaders, they were leftovers. And that left 100 rats not so long after that. And then it cost $5 million to get rid of the last 100, which cost about $50,000 a rat. So why didn't they interact in the first place? Probably because once you reduce an animal population really low, there's food everywhere. Why are they going to take the food you're offering? That's the kind of idea. So it is a costly business. So again, it's the idea of animals weighing up their options here. We try to give them the best attractant, but the device is too dangerous. And if the missed opportunity costs are too high, they can get whatever they want anywhere else, they're not going to interact. So this set of experiments was then playing around with missed opportunity costs in the form of phantom decoys to see whether or not we could tip the balance to make the attractant seem a little bit more worthwhile by changing the animal's missed opportunity costs. At least that's what we think was going on. Now, what are phantom decoys? You experience phantom decoys all the time. Marketers are using this on you all the time. I took the liberty of looking at the wine list at Tamarind restaurant in Cairns. Wine lists are my favorite thing of phantom decoys. I often go to restaurants and go, why are there $590 bottles of wine on the list? Who's buying $590 bottles of wine to eat at this restaurant? Well, it may not be there for the value of the wine. What it's doing is making you think that the $65 bottle of Jim Barry up the top there, which costs about $12 to buy, is actually not that bad of value. It's called price anchoring, and it's just changing the relative our perception. A $10 bottle of wine, a $30 bottle of wine compared to a $10 bottle of wine seems pretty expensive. Compared to a $50 bottle of wine, seems quite cheap or relatively good value. That's what a phantom decoy is. And it's used everywhere. You've probably gone to websites, online shopping, and you go, why are all these items still there that are sold out? They're there to make you want the stuff that's available more. And it's working on your perceptions of missed opportunity costs. Again, it's called the sold out effect. It's called price anchoring. But what it's doing is changing the relative decision about what the value of the things that are available that are there. There's lots of work on this in behavioural economics, where they're trying to work out why they work. There's a myriad hypotheses of why they might work. We were just trying to work on the idea that it might change the animal's perception of what it can get and the value of it, this sold out effect, working on what they're missing out on. Show them something that they really want that they're missing out on, and maybe they'll go for the thing that they didn't think was so good a little bit more. So we were seeing, can these phantom decoys or these decoy misinformation, can it increase trap success of pressed rats? And then can it increase trap success of rodents? So here we were trying to get them to interact with tracking tunnels a little bit more. We over on the left-hand side, there's a camera pointed at one of our devices. And I think, I'm not quite that tall, but you can just see a cappuccino shaker on a stake in front of the device. It had a peanut butter bake ball in it, which the rats really do like, but they couldn't get access to it in this case. And we wanted to see whether or not that would encourage them to go into the devices, which I showed you before, they really don't like going into. We used 2 types of attractants, a lower quality one and a higher quality one. It was a jam sandwich or just a sandwich without the jam, and to see whether or not the decoy, which did nothing more than be unavailable to them, could encourage them to go into traps a little bit more. And we did this at 270 sites around Sydney. When we were able, and many of them were within 5 kilometres of where we lived because it was done during COVID. So we used our camera footage to see what did the rats do when they went there. Did they interact with the tunnel? Did they sit on top? Did they make a partial entry, or did they go all the way? Did they interact with the decoy? How do they interact? Did they just sniff it? Did they actually interact with it? Did they express frustration as much as we can tell from watching a rat on a video? And it did seem to work. It did seem to change their perception, at least of the lower quality bait. So in the figure here, we've got where there was no decoy in the blue or where there was a decoy in the grey. This was the low quality bait devices. This was the high quality bait devices when there was no bait at all, or where there was just a decoy in the tunnel. And you can see that there was no effect on the high quality bait, but for the low quality bait, when the decoy was there, we had way more interactions. And this is kind of typical of what was going on. The rats would go to the phantom decoy that's in the shaker there, they'd sniff it, get a little bit frustrated, and then more often, they would go into the device. And typically, the ones that when they did go in had interacted with the decoy prior to entering the tunnel. So it was reasonably encouraging that it did seem to work. They did seem to visit those devices and enter the devices faster too when the decoys were there. This is just looking at the night that they entered, low quality bait compared to low quality bait with a decoy, whereas high quality bait, they were still going in. We possibly offered them too much of an attractive bait there, but it does seem to change their perception. There was nothing else there. We had a control for the attractant effect, and this was just whether or not they went in. Just that thing that they couldn't get access to made them go for the bait that they didn't seem to want. The last example then is, can it increase the trap success of mice? Oh, this one's got sound. This is a real problem for monitoring mice numbers. CSIRO use trapping to try and estimate mouse numbers to predict plagues. They know that they can't get all the mice that are there. They use pretty similar traps to this, so they're keen on knowing whether or not there's something that can improve trap success for mice. So we did the same kind of experiment. We didn't have as many cameras available this time. We just wanted to look at trap success. But again, we used the shaker out the front, Elliott traps with wheat as the bait, cameras set up on some of them, and the peanut butter bait ball inside the device. We had our wheat as the attractant with only wheat seeds. We had wheat with a decoy at the trap entrance or a decoy inside the trap to try and control for any attractant effect. And again, done over lots and lots of sites. Yeah, so again, our decoys did seem to improve trap success. We had a doubling of trap success almost for the rats, and this time was an almost doubling of trap success compared to the plain wheat bait when the decoys were outside. There was obviously some kind of attractant that was going on with the decoy in the trap. We couldn't tell that these were showing up and not going in, so because we didn't have as many cameras on, but we did end up with more mice being trapped. So in summary, again, these information decoys, they don't do anything, we think, apart from change the animal's perceptions of its missed opportunity cost. We're showing them what they're missing out on, and that's changing the way that they value what we're offering them. We think that these phantom decoys do work by changing missed opportunity costs. And so we have been trying to use this concept to catch the uncatchable rats. I'm not offering any promises. If you've got rats or whatever that you can't catch, you can try this, but it's devilishly hard to show. I did have a PhD student who was trying to do this, but when you're trying to catch animals that are uncatchable, it's really hard to work on. And she was trying it. So I think that misinformation is an untapped potential in wildlife management. The stuff that I've shown you is all non-lethal approaches. We don't necessarily want to, farmers don't necessarily want mice, but they don't really want the impact that's caused by mice, and they really don't want to use poisons either. And similarly, we don't want wallabies causing damage, but we don't want to be shooting wallabies and losing what positive things they do. So it's potentially a much better alternative, and it's good for native species. We think that it works better than some of the old approaches where you're trying to deter an animal because it works with animal motivations. I was talking with Annabelle on the way over here about someone wanting to know whether or not an ultrasonic deterrent would work. Well, it may work, but the problem is if the animal's motivated to get what you're trying to deter it, it's going to struggle. Whereas these kind of approaches are working with animals' motivations. What they already want to do, we're just nudging them away from that by using misinformation. It's using fake news, which manipulates us pretty well, so there must be something in it. It's really acting on the direct source of problems rather than indirectly by killing things and hoping that it will reduce things. And we think that it's a kind of missed opportunity cost if we don't use it. And so we think that it's got lots of potential in all sorts of scenarios to manipulate. Thanks so much. I'm sorry I rushed through the first part. I was worried about running out of time, but I'm happy to answer any questions. Yeah, but thanks so much for listening.
Okay, thank you, Peter. Really interesting talk. Heaps of interesting stuff there. Questions here or online, Sue?
Thank you. Really enjoyed that, Peter. And I'm going to trial one of those things, I think, in some of my research. But I did wonder, so with the decoy, I trap rodents here on campus, take students out to catch rats, and I'm thinking that the decoy just also potentially saturates the scent in the space by adding extra scent.
Yeah.
So it's got that potential to sort of bring in animals from further apart, further away.
Yeah, with the mice thing that could have happened, but with the rats, we know that it was what we were showing there was the percentage of visits that turned into interactions. So it was actually we were trying to with the camera footage. They showed up. Definitely, they showed up, but even without it was the fact that they changed their mind. So it was the percentage of visits that turned into interactions that counted. So it wasn't solely an attract and effect. We think with the mice we're not as sure because we didn't have cameras on there, and there's no doubt that animals would show up and they'd be attracted to it. But because we saw that it was the showing up but going in was higher, that's why we think that it worked. That why it worked there.
Well, and I was thinking when you had the low value bait, if you had them entering with low value bait, then you've already got them there so that shows that they'll still go looking for more food, even if it's not what they would intentionally want. Without the decoy.
I think, yes, maybe. It's in review at the moment. We haven't seen the second lot of reviews. So we'll see. I thought that might have been going on with the mice, but we don't think so much there. We'll see what the next reviewers say.
Other questions?
Hi, Peter. My question is about population. I know you mentioned how difficult it is to measure populations in these scenarios, but I wonder what would be your opinion on if you're using these misinformation techniques to deter some populations in a certain area. Will that in the long term or intergenerational time be able to reduce the population of those mice? Or are you actually just deterring the mice into a different field and they will be able to keep the population? Or are you actually reducing the resources that this population could have and therefore in the long term reduce the population size?
Yeah, that's a really good question. So what happens with the mice that are there? They just keep eating what they're normally eating before the farmers put the grain in the ground. So they're eating spilt grain, they're eating bugs, they're eating anything that they can. Will the inability to access this particular, what's sown at that point of time, will that impact the mouse populations? Probably not that much. It's a small pulse of food for a short amount of time, and it's not that much compared to how much spilt grain there is actually on the paddock. For some reason, they still dig for it, but it's not as much because there can be an awful lot of spilt grain on the paddock. Does it push them somewhere else? No, I think they just will stay there and just keep eating what they're eating because we've either shown them pursuing that cue is not worthwhile before the grain goes in the ground, or we make it unprofitable so that they just don't access this extra thing that's there for a short while. Is it going to have a long-term impact on mouse numbers? No. Mice numbers go up and down with the weather, really, partly. When we don't really know why mice plagues happen now, prior to conservation farming, they happened when there was 2 years of good rain that allowed a pretty good crop and the right soil conditions for a population to boom. We're not as 100% sure why it happens now, but populations during a boom time usually crash anyway. They either run out of food or some disease runs through them and wipes them all out super quickly. The problem is that is just prior to when the crop's usually been sown. So I don't think it has any impact at all ultimately on mouse numbers. That is the thing. I mean, the mice will still be there. Like I said, farmers don't want mouse plagues. They're happy with mice being there. They would rather not use poisons if they didn't have to because they're nasty, and they do kill birds. But it's not going to stop a mouse plague. And poisoning won't stop a mouse plague either.
A question?
Sorry.
Yeah. Any online questions? And I'll just ask one, Peter. So you sort of touched on this a little bit, but I'm assuming others have used, whether they're seeds in a field or eggs that you want to protect or whatever, have used odors from, say, predators or just noxious odors. So you sort of talked about, nudging their preferences, but these sort of straight up deterrence things seem like they would be really efficacious if you could, for instance, treat your seeds and that they would smell really bad for 2 weeks and nothing would eat them. But so have people, I'm assuming, have done that?
Yeah, heaps.
Heaps.
Yeah, and they don't, it lasts in short term, but hungry animals are really hard to deter, particularly when the threat's not real, when it's not realized. They habituate, like, things that, well, you have 2 ways of habituation, like a perception habituation, like this threat's not real, so why am I going to worry about it? And then there's the sensory habituation where the senses just get flooded with something and then you just smell through it, like someone who wears too much perfume. They don't smell that they just keep spraying on more and more because they can't smell it anymore. So there's those 2 ways by which it's a real challenge, and as well as the animal's motivation. Hungry animals will hunger first, and they can smell through our things probably too if they're super hungry. But if they have an alternative, it will work. So we did trials years ago trying to stop wallabies getting seedlings using artificial dog urine, and it did work, but it did shift the problem to somewhere else. But that's if they had somewhere else. When you don't have something else, and that's the missed opportunity cost idea, it still won't work because that's just kind of like the danger part of the equation. But if the missed opportunity part is still too high, it won't work. And that tends to what happens. And so they work in the short term, but they just don't have a long-term efficacy. Yeah.
I've got a bit of a follow-up question there with populations, or also what Bill was asking with the lingering effect of these measures. And I saw that most of your graphs were included, like they were a function of time on the x-axis. And I remember one there with the refuge by association, was it called?
Yeah.
You reckon you could bring one of those graphs up?
Yeah.
With the visitation of seedlings, that was 1 at the start, and then it sort of tapered off over time. A bit before that, I think it was.
Yeah.
But I think a lot of those graphs were sort of a function of time. Does that mean that the efficacy of those measures just reduces, or like goes? So over time, proportion of seedlings remain unbrowsed is 1, and then over time they get used to the deterrent and overcome it and still browse anyway?
Like it can do. This works best when it's a pulse thing, like a short, like a breeding event.
Yeah.
This is a bigger challenge, absolutely, because animals, like they learn, like they Bayesian update. The risk is here that they actually will learn that the smell of this plant that they think they don't like actually means something that they do like. That could definitely 100% happen, and that's what we're going to try. That's what we're trying in Tasmania. Does this work longer than here? So those ones probably, were just lucky that not lucky, but the treatment did work, that they survived. But would they need to last a lot longer than 65 days? Whereas with the seedlings, the wheat seeds, they've only got to last two weeks, if that. Birds' nests, they've only got to survive 3 weeks. So longer term things are a real challenge for the very reason why we know this kind of works, in that animals are Bayesian updaters. They will update their knowledge based on experiences, and they could learn new associations. Yeah.
Hi, thank you for the talk. I really enjoyed it. My question is, do you think the shortcomings of like deterrent odors would apply to your experiments when you saturated the environment with positive smells? Do you think those would also be a short-term limitation?
Well, we think that the deterrents have a shortcoming because animals habituate to them. Whereas what we want them to do in this case with these odors is habituate to them. We actually want them to go, nah, it's not worthwhile. So, that's why I think that it works with the animal's motivations. They realize that it's not rewarding, so they'll ignore it, or they realize that the deterrent is not actually there's no threat there, so they'll ignore it. The challenge, though, is whether or not they start to learn, going back to that point, can they smell through it? Can they work it out? And these only work if the animals have a plan B. So that they need to have an alternative. And for generalists, they generally do. And so it was quite surprising that for the mice that it did work, but it's just that they did have spilt grain that they could eat on the surface.
Any other questions online? Okay, all good. I'm just going to ask one final question, Peter, just leading on from this, what you talked about with the learning association and the rapid learning in the rodents. Is there some magical chemical that you could spray on seeds that would make them taste really bad for like 2 weeks, whatever, then eventually would go away? Because you talked about the learning. That might be.
So it's quite funny. Farmers saw our work on the wheat seeds. And one farmer's son had a year 6 project they had to do. So he did our experiment. It's really nice. But he added that treatment. He put on capsaicin, which is cayenne pepper, on the seeds. It has been done elsewhere, actually, and it has kind of worked, but he did it on his dad's farm, mum and dad's farm. And it did work. They did eat them less. We're working with Cyro. We wanted to do that this year with canola, which is a way more valuable crop. And one of our trials was to coat the canola seed in capsaicin, basically following on what this student did. The student was going to be on the paper, produced. However, the mice didn't dig up the canola seeds. The extra part of the story is that they were digging up the seeds once they germinated, and there was something in the germination that's giving away the seeds to them. But anyway, yes, 100%. So Cyro want to see whether or not capsaicin on seeds can be a simple thing that actually, and then maybe even using it beforehand to teach them to make that learned association. I think there's a lot of opportunities it's called conditioned taste aversion, which is used quite a bit. Again, it's hard to make it work when the animals are super hungry. Like conditioned taste aversion's been tried with foxes and eggs to try and make the eggs taste bad, but the foxes eat pretty much the most disgusting things going, and so they might eat it once, and get sick, but then they'll go, oh bugger it, I'll do it anyway, and so it hasn't really worked. So it's again, knowing what their motivations are. But I think with mice, given that they will have a plan B, it's a good option for them. Yeah.
All right. If there's no further questions, Peter, thank you. Really interesting talk. Cut across a lot of disciplinary fields, and I think people here really found it interesting. So thank you very much.